may I welcome you all with the best wishes for 2024. I hope 2023 was good for you, and this year hopefully we'll have better academic sessions, and I wish you all best of health, prosperity, and of course to your families, and high academic achievements are also part of my wishes for all the audiences, participants, facilitators across the world. So greetings from London. Good morning and salam alaikum to all. And this is the first webinar starting in 2024, which hopefully will be the first raindrop followed by torrential rain in the next 12 months or so. So here is a brief audit of 2023 before I welcome Professor Shahid Mahmood Baig to give a talk. Let me just update you that we had 80 webinars by very experts in different fields of medical education and clinical sciences. We had 2 conferences virtually and blended. We ran 4 courses that are given here with different universities. We published a book on global medical education in normal and challenging times. It is to be published by Springer Nature in March, but it's already compiled in the printing house at the moment. We had 3 workshops and rolled out online system for webinars, lectures for CPD, CME, and viewpoints, and we have a very advanced LMS system which is developed by Dr. Moshe Iftar. We also see collaboration with 4 reputable universities. Can I have the next, please? In 2024, we expect that we'll expand our services. That will be our 4th year of functioning because we started in 2020 during the COVID era, and now we're getting on. So we have lined up 26 webinars of experts in clinical fields as well as medical education, research, etc. We have 5 conferences lined up, 6 to 7 courses with collaboration with other universities, and 1 more book, which is a global encyclopedia of Islamic medical ethics, which at the moment is under compilation with the help of 40 authors from 21 countries. So that will be a monumental work. Hopefully we'll publish it, we'll complete it in 2024 and publish next year. We're also looking for new collaboration with the reputable universities. So all in all, it has been a fairly satisfactory progress, and God willing, we'll progress further. With those words, I will invite Professor Shahid Mahmood Baig. And could I have Shahid Mahmood Baig's brief slide, the first slide showing, he's got so many credentials, and recently he's just retired as chairman or president and the overall in charge of Pakistan Science Foundation, and he's joining the Al Khan University as Professor of Molecular Biology and Human Genome Project in Genetics. He's going to talk about challenges and opportunities of doing science in a developing country. I mean, who could talk better than him, that he was the chairman of the Science Foundation? He has got so many qualifications, starting from MSc, MPhil, PhD, etc., 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 from different countries in the world, from Pakistan, and from Turkey, and from the United States. And of course, he's traveled extensively. He's a member of the editorial board of various scientific journals. He had 149 peer-reviewed research papers with high impact factor, collective impact factor exceeding 700. And he has trained and mentored 34 PhD scholars. I mean, can you imagine? In one single life, he has achieved so much that many people can't do even in 5 or 6 lives. He has delivered lectures at various universities in the USA, Sweden, Denmark, Germany, Saudi Arabia, South Korea, and other countries. And of course, he's been rewarded by the government of Pakistan with various medals, which are glorious medals of achievements. And I think it's Hilal Imtiaz, and Dr. Baig will tell us more about it. And recently, as I said, he was the chairman of Pakistan Science Foundation, granting various scholarships and various grants to scholars across the world, and mainly in Pakistan and the developing countries. So let's now welcome Professor Shahid Mahmood Baig, who is a professor at the Al Khan University, Karachi, Pakistan, to conduct the webinar. Over to you, sir.
Thank you very much, Professor Shabi, for the extended introduction. It's my pleasure and honor to be with you today. Thank you, Professor Shabbi, Professor Mulazim Hussain, Dr. Mubashir, all participants. It's indeed a pleasure talking to you. And today I'll discuss with you the challenges and opportunities of doing science in the developing countries. The developing country, I have taken the example of Pakistan. What are the opportunities and what are the challenges of doing science in a country like Pakistan? Of establishing the scientific institutions and bodies and R&D institutions in Pakistan. In Pakistan, the Pakistan Academy of Sciences, the idea was conceived in 1947. And Pakistan Academy of Sciences was established in 1953. And the PCSIR, the Pakistan Council for Scientific and Industrial Research, it was established in 1953. Pakistan Atomic Energy in 1956, SPARCO in 1961, PCST, Pakistan Council for Science and Technology, or National Commission for Science and Technology in 1962, HEJ and ICCBS, they were established in 1967 and 1976. The Pakistan Science Foundation was established more than 50 years ago in 1973. And the Ministry of Science and Technology, which is heading 16 organizations, NIE, PICRET, NIO, PCSIR, Pakistan Science Foundation, NICA, and there are also 3 universities, ComSats, NUST, and NewTech. These are also under the Ministry of Science and Technology. And it was established in 1973. Pakistan Agricultural Research Council was established in 1981. At the time of partition, when Pakistan was founded, there was only one university, Punjab University, which was established in 1882. And University Grants Commission was established in 1974, and it was converted into Higher Education Commission in 2002. Currently, there are 259 public and private sector universities, and there are more than 40 centers of excellence dealing with various disciplines of science, including chemistry, physics, nanotechnology, earth sciences, etc. In Pakistan, there are several challenges and opportunities. Pakistan is strategically located, the 5th most populous country of the world. It is fertile land and one of the best irrigation system. The challenges are population. A big population is a big challenge. Plus, we have the problem of food security, climate change, energy crisis, and on top of that, low investment in science and technology, health and human resource development. So these are the area which needs a lot of attention from the government sector and private sector, etc. At the same time, there are several opportunities to do good science, to do R&D, to develop products and businesses. Number one is genomics. Since I'm a geneticist, so I will talk about genomics, the scope of genomics, the research potential in the product and the application of genomics in Pakistan, the agriculture, climate change, big data, drug discovery, IT, IoT, electric vehicles, nanotechnology, and so many others. And this 21st century has been declared as a century of biology and the century of Asia. Now we look at the gross domestic product, that is the GDP of various countries. At the top, it is United States, which has over $21 trillion GDP, one of the largest economies in the world, and followed by China, the second largest economy, with $16 trillion. If you look at Pakistan, we have only a GDP of $300 billion, not trillion, $300 billion. And the GDP, as you know, is the standard measure of the value added created through the production of goods and services in a country during a certain period. Each country have apex body of science, we start with the National Science Foundation of USA. It was established in 1953, and the annual budget for science is 7.8 billion US dollars. And this NSF, it deals with the natural physical sciences and the computer sciences, whereas the funding for the health and biomedical and health-related research, it is provided by NIH, which was established in 1887. Its annual budget is $45 billion. And with this, they generated a business of US $97 billion economic activity in 2022 in biomedical and public health research. NSFC China has a annual budget of 4 billion rupees. NRF, National Research Foundation of Singapore, has an annual budget of 3 billion rupees. If you look at Pakistan, this budget is in rupees. PSF has a budget for R&D to support the whole of Pakistan, less than 1 billion rupees. Same is the case with the Higher Education Commission. We have about 1 billion rupees for R&D. Although HEC has a big amount of money every year, but it is for various higher education and academic programs, which is 59 billion rupees. And the percentage of gross domestic product of various countries for R&D, this is very important. How much of your GDP you are spending for R&D? If you look at United States, they are spending 2.7% of their GDP on science and technology and R&D. China, 2.4%. If you look at Israel, they are spending 4.9% of their GDP on science and technology. The world average is 1% of their GDP. In India, they are spending 0.7% of their GDP on R&D. But in Pakistan, the picture is not very happy at all. We are spending only 0.16% of our GDP on science and technology. And the GDP is, if you remember, it is only $300 billion. Billion rupees, not dollars. So here you can see the priority of the government here. The science and technology is not on the priority. There are several other projects which are on the priority list of Pakistan. So we have to look at the other options and opportunity to do good science and to develop other strategies to do some quality science. And if you look at the SDGs, which have been established, which has been set for 2030, and the SDGs are intended to provide a shared blueprint for peace and prosperity of the people and the world. They call the global cooperation and concerted efforts by government, business, and civil society to achieve a more sustainable and equitable world by 2030. According to these SDGs, there should be no poverty by 2030, zero hunger, good health, quality education, gender equality, clean water, and so many things. And for the low income and middle income countries, almost the major SDGs are same: no poverty, zero hunger, good health and well-being, quality education, clean water, etc. So this is what we are aiming to do by 2030. If you look at the Nobel laureate, which this region has produced over the last many years, Professor Abdul Salam, he got the Nobel Prize in Physics. He was born in Jhang, a part of Punjab, southern Punjab. He got the Nobel Prize in 1979, and then Professor Harbin Khurana, he was born in Sahiwal, Raipur, Sahiwal. He got his master's from Punjab universities. Then at the time of partition, he moved for his PhD, and during partition, he was in the UK. Then his family opted to live in India. But we own him because his education was here from Punjab University, and he moved to UK from this part of the world. Similarly, Chandrasekhar, He got the Nobel Prize in 1982, and he was also born in Lahore, although he then moved to India and spent rest of his life in India and in America, etc. So these are the three Nobel laureates which are produced by this region. Uh, but after that, we are still waiting to see any Nobel laureate or any quality of science which can compete for Nobel Prize. If you look at the funding opportunities in Pakistan, a major chunk of the funding is coming from Higher Education Commission, followed by Pakistan Science Foundation, and then there are Comstech and Ignite and some other funding agencies which are providing funding to do science and technology in this part of the world. The position of Pakistan in the global research landscape is not bad. We are publishing 66% of our publications in journal impact factors, and we are at 44 global ranking of the total number of publications and 43rd global ranking for the number of highly cited papers. It means in Pakistan, from the developing countries, we are doing good quality research, publishing in good journals. And if you look at this Pakistan's place in the global research, again, it is heartening to see that Pakistan's publications in category normalized citation is on the rise over the years. This data is up to 2019, and it gives a quite heartening and refreshing picture. If we look at the percentage of documents in Q1 journals, although we are at the bottom of, if we compare with Malaysia, South Africa, Singapore, Saudi Arabia, Egypt, Pakistan at the bottom. You see this blue line is here, and it is led by Singapore, the green line at the top. It represents the Q1 documents from Singapore. The trend is also increasing in case of Q1 journals from Pakistan. And if you look at the discipline-wise publications and the output, in case of clinical medicine, we have a very good representation, followed by chemistry and plant sciences. The less contribution is from space sciences, psychology, and neurobehavioral sciences, etc. Pakistan impact by research areas. You see in the mathematics in Pakistan, we are on the top. The world average is 1. In mathematics, we are at 1.5, followed by physics and then engineering. And again, the lowest contribution from Pakistan is in the case of space sciences, pharmacology, and toxicology, etc. These are the major areas of research in health sciences. You see, there is a lot of potential since we are a very big population. We are practicing cousin marriages. The family size is very high. Each woman has 3.5 children, whereas the world average is 2.5 children. And this population and the youth in Pakistan is the strength, and these are the areas where a lot of research can be done, and there are some very good groups which are doing quality science and trying to resolve the current scientific issues in Pakistan. The areas involve genetic diseases, infectious diseases, metabolic disorders, diagnostics, vaccine therapeutics, probiotics, drug discovery, cancer biology. And these are so many areas where there is a lot of potential and a lot of good science is being done by Pakistani researchers and scientists. If you look at the Pakistani population, what is the potential of studying genomics in Pakistani population? We are a unique demographic case, more than 225 million people. And this population can provide knowledge on evolution of modern humans, and important and useful data of a highly inbred human society, which is helpful for understanding the relationship between genetics and disease. So this is the major contribution the Pakistani population is contributing in the biomedical sciences to understand the genetic diseases, to identify the drug targets, and then to develop and to design the precision medicine for the treatment of these diseases. And currently in Pakistan, the available genetic information is being used for the counseling, prenatal diagnosis, and development of precision medicine, etc. The annual population growth rate in Pakistan is 2.3%. The world average is 1%. We have a very high fertility rate, 3.5 children per woman. The world average is 2.2 children per woman. And we have a very strong local tradition of cousin marriages, which is 70% of the marriages in Pakistan are cousin marriages. These are within the brotherly, within the tribe, between the first degree cousin, second degree cousin, and the third degree cousins. And moreover, we have limited healthcare services, and the number of genetic diseases. If you look at this map, the dark blue areas, these indicates the populations with high rate of consanguineous marriages. This blue is Pakistan, and these are some other Islamic countries and some African countries where the Muslim population is practicing the cousin marriages. And let me share with you, the cousin marriages are only allowed in the religion of Islam. In other religions and other societies, these cousin marriages are not allowed. If you look at this graph, you see this represent the consanguineous marriages in different population. In Muslims, the overall rate of consanguineous marriages is 50%. In Pakistan,
لا إله إلا الله وحده لا.
In Pakistan, the rate of cousin marriages is 70%, followed by Saudi Arabia. In Saudi Arabia, the rate of cousin marriages is 67%. But the problem in Pakistan is that our population is 225 million people, whereas in Saudi Arabia, the population is only 30 million, and they have very good healthcare system, and the economic condition is much, much better than Pakistan. So we are dealing with the very big problem of cousin marriages, which is leading to a very high risk of genetic diseases. The risk of autosomal recessive genetic diseases is 18 times higher in the cousin marriages. This is a typical pedigree which shows the result of cousin marriages. You see.
Sorry, may I request the participants to mute themselves, please? Professor Baig is giving a wonderful talk. All participants are requested to mute themselves. Sincere apologies, Professor Baig.
Thank you, Professor Shabi. Thank you. If you see, this is a typical autosomal recessive pedigree. There is skipping of generation, but at some time, in some generation, due to first cousin marriage, the affected births appear. The genetic diseases, these are mainly of three types. Chromosomal disorders, we call them rare diseases, then the Mendelian disorders, and the third is the complex disorders. In case of chromosomal disorders, the most common examples are Down syndrome, Turner syndrome. And if we look at the monogenic disorders, these are also known as monogenic or Mendelian disorders. These are caused by point mutations. These are mostly in the coding regions. And there is a little or no role of environment. The examples are thalassemia, primary microcephaly, deafness, eye disorders, intellectual disability, etc. And the third category of the genetic disorders is the complex disorders. These are also known as polygenic disorders or multifactorial disorders. These are relatively more common. They include the cardiovascular diseases, obesity, schizophrenia, cancer, allergy, and high blood pressure, etc. And these Mendelian disorders, these are caused by dysfunction at a single locus. These are inherited or familial. So far, more than 10,000 genetic diseases or Mendelian disorders have been described at clinical level, and only half of these, that is less than 5,000, have been characterized at molecular level. And this is what we are doing in Pakistan because of our large population, cousin marriages, and large family size. Pakistani population is the best population to study and characterize these diseases at molecular level. And unfortunately, for most of these genetic diseases, there is no treatment. So what we are doing on the basis of this molecular and genetic knowledge, we are providing the prevention and genetic counseling, and treatment is only possible if a disease is well characterized at molecular level. So for finding the disease, some of the diseases of these 5,000 or 10,000 diseases, these are curable. But for that, we must have the genetic knowledge of these diseases. So what are the genetic variants we are looking at and how we can treat them? There's a supplement, protein supplement, alternate enzyme. Now we have gene editing system, the CRISPR-Cas9, whether these variants can be corrected by the gene editing. So for that level, we have to do the molecular diagnosis and molecular characterization of each and every genetic disorder. In Pakistan, there is no nationwide registry. The incidence of most of the diseases is not known. Difficult to access the affected families. Most of these families, they live in the rural areas, poor disease awareness, limited access to the healthcare facilities, difficult to diagnose due to rarity of these disorders. This is common throughout the world. Genomic research, it is challenging and expensive worldwide. The next generation NGS it is cost effective comparatively. It is time saving. There are few academics groups in Pakistan who are using these NGS technology, and these groups, they mostly rely on international collaboration. As I mentioned, this is a very expensive science. It needs a lot of money, infrastructure, the teamwork, etc. So Pakistani institutions cannot work in silos. We have to establish the collaborations and research and networking to work on these very challenging genetic diseases. I give you the example of thalassemia from where we started in Pakistan. Thalassemia is the most common genetic disorder in Pakistan. The allele frequency is more than 6%. It means more than 12 million people of Pakistan are carrier of thalassemia. And each year, there are more than 5,000 affected children which are born, and the affected children, they require regular blood transfusion to sustain life. The treatment in some cases is possible. It is bone marrow transplant or stem cell technology, but it is very expensive. Each bone marrow transplant, it costs around more than 3 million Pakistani rupees, which is beyond the affording capacity of most of the families. So we adopted a model. We have a sister organization in Multan, which is known as Minar. We selected the trials. Trials mean the patient and the parent. And we characterize the most common mutations which are causing the thalassemia in Pakistan. Luckily, there is a set of 7 to 9 mutations, most common, which is responsible in more than 80% of the cases for causing the mutation. And these are done by a simple arms PCR method, which we developed here. And in case of rare or novel mutations, the Sanger sequencing method is used. And to these couples who have at least one affected child, we are providing the first trimester prenatal diagnosis through chorionic villus sampling. And we are doing the cascade testing. Cascade testing is, for example, this large pedigree was identified by this affected child, this index affected child. When we screen the extended pedigree, you see there are so many affected, and there are so many children who die undiagnosed. Actually, they were having the beta thalassemia major. We provide the prenatal diagnosis through chorionic villus sampling. So far, 1,500 couples were provided the prenatal diagnosis, and you see the result of this prenatal diagnosis. This is typical autosomal recessive disorder result, typical Mendelian ratio. About 25% of the fetuses, they were found affected. That is, they were homozygous for the parental mutations. And in Pakistan, we have the fatwa, or the religious verdict from the ulama, that if a child is suffering from a serious disease which is not curable, then the termination of pregnancy is allowed in the first 120 days of the pregnancy. So since these families, they cannot afford the bone marrow transplant or the stem cell technology, and they have the experience of raising a child with beta thalassemia or any genetic disease. So these families, they opted for termination of pregnancy. And this green area, it shows the carrier are heterozygous like the parents. And this yellow area, this shows the this yellow is the heterozygous or carrier, and the green are the normal, which don't have any mutation. This is a scene from a thalassemia transfusion center. You see the cost every year on the management of these more than 7,000 children who are born every year, and the number of thalassemia children who are transfusion currently, it is more than 50,000 in Pakistan. So they cost more than $120 million per year in Pakistan for the management of this single disease only. We are trying to develop a novel therapy for hemoglobinopathies. It includes the thalassemia and sickle cell anemia. We are doing this project at Aachen University, and hopefully we will be able to give a cost-effective therapy to boost the fetal hemoglobin in these children. For other, thalassemia is a single gene, monogenic, relatively simple disorder, although it is fatal and life-threatening. But in terms of genetics, it is considered monogenic and simple genetic disorder with a few mutations only. Whereas the other Mendelian disorders, they are not that simple. It is very challenging to work on these Mendelian disorder. If you look at the Joubert syndrome, you see there are so many genes involved. A single disorder and so many genes involved. So diagnosis and screening is not as simple as thalassemia. So we need the high throughput system and sophisticated technologies to work on these very challenging Mendelian disorders. So one disorder with multiple genes. And there is another situation where we have a single gene, for example, the TMEM67, it is involved in causing so many genetic diseases: Joubert syndrome, Meckel syndrome, Cow syndrome, Coloboma, Ryan syndrome, Bardet-Biedl syndrome, nephronophthisis, etc. So we face various challenging situations while working with the genetic diseases. And these are least characterized in Pakistan. The scope of computational biology is also very important. It supports the drug designing, the genome assemblies, the biomarker, gene regulation, modeling, simulation, proteomics. and so many other areas where computational biology and biophysics is supporting all these biomedical sciences. And there are some very good groups who are working in Pakistan to support this biomedical sciences. We started this work at National Institute for Biotechnology and Genetic Engineering in the Human Molecular Genetics Lab. It is under the Pakistan Atomic Energy Commission. And you see these families who have genetic diseases, some of them, they live in the cities, but most of these families who have multiple number of affected in their families, they live in the remote areas, in the rural areas. They don't have the access to the healthcare facilities or the hospitals, or if they visit the hospital, they are simply told that, informed that this is a genetic disease, it has no treatment, so simply avoid the cousin marriages if you want to prevent this disease. But this is not a solution. They need proper genetic counseling and proper clinical investigations and genetic investigations to provide them the carrier screening tests so they can identify. There are many families who cannot avoid the cousin marriages. So we have to provide them the carrier screening test. And that's what we are trying to establish, to establish the carrier screening test for all the diseases which are found in Pakistan. You see, these are in a number of different diseases which we identified from various areas of Pakistan by conducting the field sampling trips. And for further genetic diagnosis to finding their disease gene, which is involved in the disease gene, there is certain criteria to include the families or the pedigrees. The parents has to be first or second or third degree cousins. They should have 2 or more than 2 living affected children, so we can validate our data that this is the gene and this is the variant which is actually causing the disease. So in this way, we collected up to 2020, we collected more than 3,000 such families, and we started working on them. These are some of the pictures of the field sampling trips which we conducted in various areas, remote areas of Pakistan, Kashmir and KP and Punjab. We also moved in Sindh and Balochistan, but most of the time we focused on Punjab. Punjab, is the biggest province with more than 50% of the population. And in the southern Punjab, there are many rural areas, remote areas who are practicing cousin marriages, and they have a lot of sick children who need these clinical and genetic diagnosis. So these are some of the pictures of our field sampling, pedigrees, and genetic counseling sessions, etc. How we work on these diseases. As I already mentioned, genomic work is very challenging. It needs a lot of money, expertise, infrastructure, instrumentation, sophisticated techniques. So we are doing some of the work in Pakistan, identification of the families, genetic counseling, and prenatal diagnosis if the disease is characterized, sampling and clinical diagnosis, then the linkage analysis, diagnosis of the known mutations, and then we look towards our collaborators for the whole exome sequencing. Whole exome sequencing is very expensive. Each sample, it costs at least $250. So we cannot, since we are doing this on research basis, we are not charging anything from the patients. We are providing the prenatal diagnosis and the screening absolutely free of charge to these families who are participating in this research program. Similarly, the whole genome sequencing, again, very expensive. We do it in the collaboration with our collaborator in Sweden, in Denmark, in USA, in Germany, in Switzerland, in China, and other countries. And the role of collaboration, you see, it's very important. The double helix DNA in 1953 was discovered by collaboration of Watson, Crick, Wilkins, and Franklin. They met in 1951 They were from different universities and different areas, but they collaborated, and ultimately they discovered that DNA is a double helix. And the theory of relativity, we think that the theoretical physicists, they work in isolation. But you see, these scientists like Einstein, he collaborated with his two students, Marcel Grossman and Michel Bessot. And at one time, Einstein told Grossman, "You must help me, or else I'll go crazy." So you see how much he was depending on collaboration and on his team to find the novel things in physics and present the theory of relativity. And there are some very big projects. For example, the global National Geographic, it is being run by IBM and Great Foundation, and the DNA sample across the world to map their ancestry and migration patterns. 140 countries are participating in this project. So this is how the collaborations and the networking is important to share your resources, your expertise, your knowledge, your data. And you see, this is our collaborative network, our consortium of genetic diseases. We established collaboration in Germany, in Sweden, Germany, Denmark, Germany, China, France, USA, UK, Switzerland, USA, Germany. And there are several other collaborators who are not seen in this picture. They have different expertise in genomics and different lab facilities. Everybody is expert of something, and we are all working as a team to work on various genetic disorders. And so far in the last 15 years, we have found so many novel genes, characterized them, found the variants, and some of these diseases are curable. And many of these will be curable through gene editing and some other techniques in the coming 5 to 10 years. Let me give you some examples of the genetic diseases which we have characterized here in Pakistan. You see, this is the example of primary microcephaly. In the local language, we call them shadole, chue, ya rat people. So these are actually, this is a neurodevelopmental disorder, which is characterized by head circumference of an affected individual is less than 3 standard deviation below the population age and sex, and it is associated with non-progressive mild to severe intellectual disability or mental retardation. You see, this is a normal head circumference, and this is the affected primary microcephaly. Here you can see the difference in the cerebral cortex. This is the normal head, and this is the reduced cerebral cortex here. This is the MRI of patient brain. You see reduced cerebral cortex, and this is a control, a normal human brain. And this primary microcephaly is associated with reduced head circumference and mild to severe mental retardation or intellectual disability. You see, these are the type of pedigrees we identify in the field. You see, this is one family, a pedigree of one family, highly consanguineous, very simple or uneducated family living in a remote area in Punjab. And you see, there are so many affected children in this family, and they don't know that this is a genetic disorder and what is the role of cousin marriages and how to avoid or how to prevent these genetic diseases. So we work on these primary microcephaly. The incidence of primary microcephaly is highest in the world in Pakistan. It is 1 in 10,000 according to one report, but according to my own experience, it is 1 in 5,000 in Pakistan. There are so many affected children in this region. So far, 25 genes have been reported which are responsible for causing the primary microcephaly. And 6 out of these 25 genes have been reported from my lab. These are the genes K14, SAP135, KNL1, NUP37, SAS6, and RRP7. These are the genes. When you find a gene for any disease, it is considered as a high achievement in biomedical sciences or genomic science. So this is a great contribution which Pakistani population is contributing in the biomedical science for the whole humanity. At one hand, it gives us the future targets to treat these diseases in future. And on the other hand, it gives us the understanding of the human brain development, about the evolution, why we are human and why the monkeys are monkeys and the gorillas are gorillas. So this is the knowledge which make us understand the human evolution and brain development, and at the same time, diagnosis of these diseases, what are the genes and the variants which are involved in the pathogenicity of these diseases, and whether these genetic variants, these are curable, these are editable in the future. So thanks to the next generation sequencing, you see the first human genome was completed in 13 years. It was started in 1990 and completed in 2003 with the cost of $3 billion US. But now it can be done with the cost of $500, and overnight it can be done. So thanks to the next generation sequencing, the cost is coming down, but still our population, our general people, they cannot afford even $500 or $250. We are trying to further reduce the cost of these genomic technologies so our population can be beneficiary of it. This is an old sequencer. It is still in practice in the big lab, research labs and big diagnostic lab. And now we have the Oxford Nanopore. Now the clinician can have this USB stick size genome sequencer in his clinic, and he can do the whole genome sequence or whole exome sequence overnight in his clinic. You see now these are the diseases which are not diagnosed, and the children were dying undiagnosed. These are some metabolic disorders. This is an example of thiamine metabolism dysfunction syndrome. You see the child is brought in the hospital, presented on the first day. On the second day, the blood sample is taken, sequencing started, genetic results on the second day. On the day 3, the genomics and medical genetics expert, they discussed, diagnosed, and started the treatment. And the day 3, the treatment started. And on the day 5, the child is released from the hospital, discharged from the hospital in a healthy condition. So these are the benefits of whole genome sequencing and next generation sequencing. So on the basis of these, our research, which we conducted in Pakistan, we published our results in the high impact journals, in American Journal of Genetics, in Annals of Neurology, in Nature Genetics, in Journal of Clinical Investigation. So these are different diseases, starting from microcephaly to deafness and other diseases. These are some of the mechanism and functions of relevant protein which we described, that these are the genes, these are the proteins, and this is the function which is causing this disease. There was one family with profound deafness. They cannot hear or they cannot speak anything. But in this family, there were sudden deaths, multiple sudden deaths. The deafness doesn't lead to sudden death. When we extensively studied this family, we found this, the mutation in CACNA1 gene, it is also causing the bradycardia, the slow heart rate, which leads to the sudden death. So on the basis of this, we advise the family to change their lifestyle, to have the regular ECGs, etc. Now there is no more sudden death in this family. This paper was published in Nature Neuroscience in 2011, and it got the best paper of the year in Germany and got the Gears Foundation Award in 2011. There are some other examples. For example, generally the cancer is considered as the multigenic or complex disorder, but there are less than 2% of cancer cases which are caused by germline mutations, where the mutations are inherited in an autosomal recessive manner. We were referred a family where there were so many deaths with brain tumors. We embarked on this genetic diagnosis, and we ultimately found that there is a mutation in PMS2 gene. This gene is involved in the DNA mismatch repair mechanism, and a mutation of this in a codon, it leads to a stop codon and leads to brain tumor. So on the basis of this, we established the prenatal diagnosis on the basis of this mutation and provided the prenatal diagnosis to this family. 3 pregnancies were found affected, they were terminated, and the 4th one was found homozygous normal, and the baby is now almost 5 years old, and she's healthy. So this was the first prenatal diagnosis of any brain tumor by DNA analysis. Similarly, we characterize some hemangioblastoma, characterize their mutations, provided the carrier screening so they can have early risk assessment, and if they like, they can start the early treatment as well. And the other science which is now very popular is the bio-big data science. Bio-big data describes a large volume of data in bioinformatics, computational biology, and it represents a new paradigm that transforms studies to a large-scale research. Now, China is doing the big biodata science. We are part of it. And the future medical science will depend on this biodata, whether it's a virus, some animal, a plant, or human. These laboratories in China and in some other countries, they are generating the big biodata, although at this stage, it is the investment, but it is the future treasure. All the treatment, prognosis, and early disease risk assessment will depend on this big biodata. And whoever will have this big data, they will lead the world. Currently, China is having the largest big biodata of all the living things. So to me, China will rule the business in biomedical in future. So these are different applications of big data in diagnostic, in preventive medicine, in precision medicine, in population health, etc. And you see there is a growth in healthcare sector. It is more than 10% than manufacturing, financial service, media, and global data sphere, etc. Presently, the healthcare industry generates around 30% of the world data volume by 2025, and it will be at 36%. So these are some other results of next generation sequencing. You see, after the introduction of next generation sequencing, different sequencing increase many folds. This is a picture of China National Gene Bank. We are collaborating there, and they are sequencing each and every living thing. And I must tell you again that they will rule the future health sciences, because they will have the whole big biodata of all viruses, how the virus will behave, which vaccine will be effective, which drug will be most efficient. It will depend on this big biodata. And these are the next generation sequencing platforms which are sitting under one roof in BGI. These are 150 platforms, and they are working day and night. I took this picture 6 years ago, but now the number of NGS platform has multiplied in this institution. We are also trying to establish a similar genomic research center, National Genomic Center in Pakistan. We are working on it to do the disease gene identification, genetic profiling, carrier screening, prenatal diagnosis, pre-implantation genetic diagnosis. In some countries, like in Saudi Arabia, the prenatal diagnosis is not allowed because the termination of pregnancy is not allowed after 7 days of the conception of the pregnancy. According to Saudi ulama, the life start at 7th day of the pregnancy. So in Saudi Arabia, they are practicing the pre-implantation genetic diagnosis, where the prenatal diagnosis is possible in the first 7 days of the pregnancy. But in Pakistan, we are trying to establish it, but it is very sophisticated. We are having some problem. So this is the outline of the genomic center. It will be doing the genomic profiling, genetic counseling, genomic services, and genomic research. And I tell you, Pakistan and the other countries who are a big population, who are having cousin marriages, or whether they are having cousin marriages or not, each and every population has the genetic disease. There are some very good genetic centers which are established, the national genomic centers established in various countries, but in Pakistan, we are the most needy population, most deserving population to establish such a center, and we are trying to establish it. We have already done some groundwork. And from the Pakistan Science Foundation, where I was the chairman till last week, we provided funding during the COVID-19, and ventilators and catheters were developed by NUST and Rifa University, and these are still under the approval process by TRAP. In Pakistan, another challenge is we have the very minimum budget for science and technology to support the research in Pakistan from the government public sector. We are trying to connect the industry and academia as in the developed world, the industry is supporting the academia for the research and solutions to the problems. We are trying to establish such linkages and such PPP, the private-public sector partnership, and triple helix or consortium research projects to promote and to popularize the science and this networking in Pakistan. We have also introduced the venture capital in Pakistan. We have many young innovators and the startups who need some investment. Now we have the venture capitalist who visited Pakistan from UK, and they have a US dollar 100 million for Pakistan, and 10 million for agriculture, and maybe 20 to 30 million for biomedical sciences. We are trying to make some protocols and some rules and regulations, some registration, etc. It is neither grant or loan, it is investment, and you see the businessmen are very smart. They only invest in potential projects. This is my team, which work with me. These are mostly the PhD and postdoc students. Now I'm no more at NIPG or at Science Foundation. These students of mine, they are working in Pakistan and abroad, and we are still part of this research network, and we are working together to characterize the genetic diseases in Pakistan. And these are our collaborators. You see, the collaboration, international and national, is very important. We have collaboration from various countries and funding from Swedish Research Council, Uppsala University, Lundbeck Foundation, Higher Education Commission of Pakistan, Center for Molecular Medicine, Cologne University, Germany, and several others. So I must emphasize that it is a collaborative work. You need a lot of expertise and a lot of facilities, a lot of funding. So nobody can work in isolation. And this is one of the pictures which I took during my early research. It is one family. These are actually the cousins of a pedigree. They are suffering from a skin disorder. I thank you very much for your patient listening. I hope I was able to convey the challenges and opportunities. There are several challenges, but at the same time, there are so many opportunities to do very good science. There's a lot of potential, as I mentioned, in genomics and other disciplines of life sciences, agricultural sciences, nanotechnology. What we need to do, we have to open our doors and establish the networking, the collaboration. We don't need to be secretive. A few years ago, we used to be secretive. We were hiding our data and the results from other, but now the world has changed. Whenever we find anything new, we put it on Gene Matcher and ask others to join us. If they have the same type of results, we can join together and we can publish and we can generate some real good, genuine scientific data, which is ultimately beneficial for the treatment of the diseases, diagnosis of the diseases, and for inventing and discovering new and more efficient drugs. Thank you very much for your patient listening.
Thank you, Professor Baig. It was a wonderful, wonderful top-class lecture, as expected of a scholar of your caliber. You have the experience not only as a basic scientist, but also supervising and granting funds to all the various participants who are working in Pakistan and elsewhere in the country. Collaboration is the answer, and definitely collaboration is needed. I'm just reading this book written by Siddhartha Mukherjee. It's titled as The Gene. And after listening and watching your slides, I'm convinced that the future of medicine and biology and biological sciences and plant biology and everything is dependent on the identification of genes and the human and the global genome project that you talked about. Now, I have a few questions that people have asked. Let's start with Professor Farzana Nasir, who was the professor and head of genetics in Karachi, now settled in Canada. And she has asked this question. I'll just read it out to you. Are you working on the screening of thalassemia amongst carriers? Are you working on screening of thalassemia amongst carriers?
Yes, we are doing that. We started this program 20 years ago, and definitely Pakistan is a very big population. We cannot screen the whole population. We don't have the resources or the funding to screen the whole population. What we are doing, I have shown one slide of cascade testing. Whenever we identify any child with the transfusion-dependent beta thalassemia, what we do, we screen the whole extended family, their cousins, their aunts, their uncles, everybody in the extended family. And in this way, we identify the carriers in that extended family. We provide them the genetic counseling at the same time. Some of them are carrier. If they are carrier, we ask them to, if they are not married, to get the screening of their potential partner as well. And if they are not carrier, then they don't need to have the carrier screening of their partner. Although in the Punjab region and in the Sindh region, the resolution was passed from in their provincial assembly. No marriage will be registered if they don't have the thalassemia screen. But nobody is practicing it. Nobody is following. We are doing as I mentioned, we are doing the cascade testing. We are providing the carrier screening only to those families who already have a sick child. So at least they have identified the carriers. In Punjab, a program, Punjab Thalassemia Prevention Program, was started 5 years ago. Now it is closed. Their mandate was to screen the population and provide them the prenatal diagnosis, etc. So they worked for 5 years, but now this project is closed. My laboratory in Faisalabad We are still providing the screening and the prenatal diagnosis free of cost, but it is only limited to the southern Punjab. The patient are identified from Multan because of the limited resources and limited capacity, we cannot screen the whole population. We cannot offer this service to the whole of Pakistan. We have offered this facility to the southern Punjab, and the rest of the population was being taken care by Punjab Thalassemia Prevention Program. I wish and hope we can extend it to whole of Pakistan. We will try to submit a PC1 to the government of Pakistan, since every year we are having new more than 7,000 affected children every year in Pakistan.
So, sir, why confine only to Pakistan? Because in this group, there are participants from India and from UK and from Iran and Kurdistan and Saudi Arabia and Africa. Perhaps you'd be kind enough to extend this invitation to all of them.
Yes, they're all very welcome. Iran has already a well-established program for screening of thalassemia, and the thalassemia premarital screening of thalassemia is mandatory in Iran, in Saudi Arabia as well. So for any marriage certificate, they must have the screening of thalassemia. And other countries who are not practicing this, my services, expertise are available. They can contact me anytime. I can guide them. I can help them how to establish this carrier screening and the prenatal diagnosis and the screening of mutations. Thalassemia is as common in India as in Pakistan, in Iran, in Saudi Arabia, in the Middle East countries, and the other. There are only few countries, maybe in Northern Europe, there is no thalassemia. But now we have the Muslim population over there. They also have the thalassemia. But in the native European Caucasian population, there is no thalassemia. But wherever there are Muslim population, they have the thalassemia. And even if they are not Muslim, in China, there are some region where the thalassemia incidence is very, very high. I visited one of the province, Shangzhou. The thalassemia in some area, the allele frequency is more than 25%.
So there are lots of compliments for your lecture and presentation, and they've requested the slides to be uploaded in the system, which we'll do. Now, I have a question of my personal interest as an otolaryngologist. There was a time I was doing a research on congenital deafness in a school called Idaru in Karachi, which is now a famous institute run by Nadira Panjwani. And one of the syndromes that we found was Pendred syndrome. And of course, there was no genetic service available at that time. I was just wondering, look at your when you mentioned about the congenital hearing loss, the congenital deafness leading to bradycardia and death. I just wonder what other forms of deafnesses could be associated with congenital heart conditions or maybe metabolic conditions. Do you have any data on that?
In our data, we have found only one condition where it is associated with congenital heart defects. There are other types of deafness conditions which are associated with other diseases. Some are known as Usher syndrome or some other syndrome which are not associated with the heart. So to my knowledge, there is only one mutation or one type of deafness which is associated with the bradycardia or heart condition.
Is there any scientific test or technology progressing towards preventing congenital deafness? Because currently it's a major problem in Pakistan and of course in other countries as well, India as well. In Iraq, I've seen a lot of deaf children. Hearing aids are expensive and it's too late for the hearing aids. In certain cases of deafness, you can't help with the hearing. You need a cochlear implant, which is grossly, grossly expensive. We did a webinar a few weeks. My question is it preventable? Like one of them is consanguinity, but any other factors?
Yes, in most of the cases, these are genetic and these are preventable, but this deafness is quite heterogeneous. There are more than 150 genes which are involved. In Pakistan, there is one gene, JJB2, which is most common. It is involved in more than 50% of the cases, but worldwide, it is so heterogeneous. There are more than 150 genes. For the carrier screening, we need to have the gene panel screening. Gene panel include all the 150 or more genes which can be responsible for screening for causing the profound deafness. But this is expensive, and there is no simple biochemical or any other test by which you can identify the carrier. We have to apply the gene panel. As I said, there are more than 150 genes which are involved in the profound deafness. So the only test by which it can be prevented, it is the gene panel testing. So only.
I'm sorry for interruption because you're a shortage of time. I'm a student of ethics and I'm very grossly involved in ethical issues. You mentioned about pre-genetic diagnosis and pre-implantation diagnosis through genetic screening. I was asked this question a few years ago by a doctor. They had found in their baby to have anencephaly and dichotomy. I think it was a problem with the trisomy 21. And they requested if an alim or Rahbar or Marja Taqlid or somebody could advise them whether abortion was possible. And that was 14 weeks. I'm sorry, 14 days, not 14 days after conception. Do you have, and you gave an answer there that 7 days allowed in Saudi Arabia. So what is the maximum limit for legal abortion in such cases? Do you have any ideas?
It depends from country to country. For example, in Pakistan, previously termination of pregnancy was not allowed. So when we put up the case of thalassemia, that there are so many children, there is no treatment, and it is misery and psychological trauma for the family, there is no treatment for it. The ulama of the religious scholars of Pakistan, I can name him Mufti Taqi Usmani. Initially, he allowed 100 days. He said, okay, up to 100 days, the termination of pregnancy is allowed. The life start at 100 days, according to his interpretation. Then we found that in some cases, the prenatal diagnosis at 100 days, we don't get enough DNA to analyze the fetal genotype. It was possible at 120 days or 110 days. We requested them that they should find some way to help those families because it is very difficult for us to diagnose at 100 days. So they studied and then they interpreted and allowed the termination of pregnancy up to 120 days. In Saudi Arabia, according to their interpretation, the life start at day 7 of the pregnancy. So they say termination of pregnancy is not allowed to 7 days. In Saudi Arabia, they have introduced, and in some other countries who can afford and who have established these facilities, they can have the pre-implantation genetic diagnosis. Whereas in some countries, the termination of pregnancy is not allowed at all. If they want to have termination, they have to move to some other country. I think in Ireland or in Scotland, the termination of a pregnancy is not allowed. So they move to UK or some other country to have the termination of the pregnancy. Now, there is another question. Some of the diseases, these are not curable at this stage. Currently, some of the diseases are not curable today. But the way the science is moving, we are expecting to have the treatment of these diseases after 10 years, 15 years, 5 years, or 20 years. And for example, the thalassemia children, now he or she can survive up to 20 years or beyond 20 years. If we allow the termination of the pregnancy and the effective treatment is found maybe after 5 or 10 years. So there are so many questions. You are right, there are several ethical questions in such cases, whether the termination of pregnancy is the rightful thing or not. So it is debatable. In Pakistan, as you know, these technologies are still very expensive. The bone marrow transplant is available, but the families, they cannot afford it. They opt for termination of the pregnancy. Whichever family contact us, they want to know genotype. If the child is fetus is found affected, they go for termination of the pregnancy. If they don't have the plan to terminate the pregnancy, they don't go for the prenatal pregnancies. Many of the couples, they leave it to God. They say, whatever happens, we are ready. And we don't want to have the prenatal diagnosis or termination of the pregnancy. So it is still debatable. There are several questions and several ethical issues which arise every day. But the life is moving, the science is moving, and we are doing whichever is possible in the given ethical values and ethical considerations, rules and regulations.
Thank you so much. There are many participants. Anybody who would like to unmute himself and ask a question? Because we have about 5 minutes more. Professor Bukhari, Professor Hassan Zaidi.
Thank you so much, sir. I will
from Iran. All illustrious. Go ahead.
Professor Shahid, thank you very much for delivering such a nice and wonderful informative lectures, and hopefully not only the Pakistan, all the participants who are sitting here, and also after uploading this lecture on the website of Zibo, the thousands of the people will be benefited from your lecture. Thank you so much, sir.
Inshallah. Thank you very much. It was a pleasure talking to you and sharing my
Dr.
thoughts.
Zarizadeh, would you like to ask any question? Because I know you're doing a lot of research in Iran, Kurdistan. Dr. Zarizadeh, are you there? Mr. Hassan Zaidi from London, would you like to ask any question? So if there are no more questions,
I would only like to congratulate the speaker with his vast knowledge, which he has given us in about 45 minutes to 50 minutes, right? Ranging from ethics to, basic science to advanced science and to the practice today. Excellent food for thought for all those who listen to it. Congratulations, Professor Shahid.
Thank you very much, Zadisa. Thank you.
So with lots of compliments on excellent presentation and the request to upload your lecture, etc., may I once again thank you formally and officially, Professor Shahid Baig. You are an illustrious scholar, and hopefully you'll come back in this year, maybe towards autumn, and give more talks. Maybe you'd like to conduct a workshop on Genome Project and all the research you're doing. We'll be happy to assist you. But in the meantime, thank you again, and congratulations for the new assignment as professor at the Aga Khan University, which is a very illustrious university in Pakistan. Thank you.
Thank you very much, sir. It was a pleasure talking to you, and I'll be happy to be with you anytime you ask me. It will be an honor for me to share my experience and thoughts.
Perhaps you can find a research project in which you help us getting the
And all the participants, please feel free. If you have any question which you could not ask right now, you can just post me, send it to Professor Shabi or any organizer. They will redirect me, and I'll be happy to respond to your questions. Thank you very much, sir.
Thank you, and Happy New Year, and wish
Happy
you the
New Year,
best
everybody.
in 2024.
Thank you, sir.
Thank
May
you,
this year
and
bring
goodbye.
more happiness and prosperity. Thank you, sir. Thank you.